Translation. Translation is the process by which RNA molecules are read to build polypeptides and proteins. Polypeptides are composed of less than 50 amino acids. Proteins are composed of more than 50 amino acids. We'll use both words interchangeably in this section. Translate means to convert something from one language to another. The process of making a protein from RNA is called translation because the language of nucleotides is being changed to the language of amino acids. The protein which will be folded, processed, and eventually carry out a vital function in the cell. 3 types of RNA. Translation requires 3 types of RNA that are all created in the nucleus: messenger RNA, mRNA, ribosomal RNA, rRNA, and transfer RNA, tRNA. Before we get to the translation process, each type of RNA will be described. Messenger RNA, mRNA. As described earlier, mRNA is the final product of transcription and carries the DNA genetic message from the nucleus to the cytoplasm. It is then used by the ribosomes for protein synthesis. Ribosomal RNA, rRNA. rRNA and additional proteins make up the ribosome. The ribosome catalyzes the translation reaction that makes covalent polypeptide bonds between the amino acids, building the protein. The ribosome includes a large subunit and a small subunit. After it makes a polypeptide, the subunits separate and are reused or recycled. Transfer RNA, tRNA. tRNA carries amino acids to the ribosome, which will covalently bond them together to form a polypeptide or protein. tRNA is single-stranded, so it can fold in on itself. It folds into a T shape. The amino acid attachment site is where the amino acid attaches to the tRNA. The anticodon loop is a 3-base sequence on the tip that is complementary to the codon on the mRNA. Proteins, words, amino acids, letters. The length and sequence of these amino acids built in the ribosomes allow all the proteins in the world to be created from only 20 amino acids. This is very similar to how all the words in a dictionary can be created from a limited amount of letters. Translation. When mRNA molecules leave the nucleus, they are met with a cytoplasm packed with all the necessary elements to carry out translation. Translation requires a coded strand of mRNA, 20 different kinds of amino acids, small and large ribosomal subunits, tRNA molecules specific for each amino acid, and ATP to provide energy for the process. Translation. Initiation. An initiation complex for a translation forms from the assembly of the ribosomal subunits and initiator tRNA, met-RNA, at the start codon, AUG, on the mRNA. When all of the components are in place, the initiator complex is complete. Translation. Elongation. The ribosome attracts different tRNAs, in yellow, containing specific amino acids. tRNA molecules enter the ribosome based on a specific mRNA codon. As the sequence of amino acids grows, the ribosome links them together using polypeptide bonds, forming a new protein. This is an illustration of a growing polypeptide chain. It has methionine, valine, and arginine. Translation. Termination. At a stop codon, UAA, UAG, or UGA, a release factor reads the triplet and polypeptide synthesis ends. The polypeptide is released from the tRNA, the tRNA is released from the ribosome, and the 2 ribosomal subunits separate from the mRNA. Polypeptide synthesis repeats until a stop codon is reached. Translation. Termination. The product of one round of translation is a polypeptide with a primary structure. It will not be a functional protein until it is folded into its final shape. It spontaneously folds back on itself as it exits the ribosome, assuming its secondary structure. Its tertiary and quaternary structures may require the help of enzymes. Once it is in its final shape, it is delivered to where it's needed in the cell. Click here to watch an animation of translation.
The hemoglobin polypeptide has 141 amino acids. 4 hemoglobin polypeptides together form a hemoglobin protein. Each polypeptide is shown in a different color here. Hemoglobin has 4 heme groups that bind to an atom of iron. Iron turns the protein red, giving red blood cells their name. Polypeptides are groups of amino acids that bond together to form proteins. Millions of copies of this protein are at work in your body right now. It brings 4 molecules of O2 from your lungs to every cell in your body. That oxygen allows your cells to carry out the reactions of cellular respiration and make ATP. Ribosomes work fast. They read the RNA instructions for the first 6 amino acids in the hemoglobin polypeptide and connected them together in 0.3 seconds. They connect about 20 amino acids per second. Ribosomes build a hemoglobin polypeptide in about 7 seconds. An E. coli cell contains about 20,000 ribosomes, 25% of its mass. Humans have about 10 million ribosomes in each of their growing cells. Humans have about 37 billion cells. That is a lot of protein synthesis.